touched several times already today the topic of going to even higher resolutions. And I want to give a real high-level perspective on that, on the work that is going on by different people, and on the plans that we are having, and also the difficulties, just to give everybody a certain perspective, which is of course biased by myself. So we have all seen the BigBrain in this workshop, of course, that is available, the 20 micron model that you can find online, and that provides you a 20 micron resolution detailed 3D view of the human brain, and has been built by registering all these sections together, as explained by Hartmut and Claude. And there was a lot of great work, like by you, Konrad, for example, to provide these beautiful layer segmentations, these are among the most beautiful images I know. Yeah, this is so nice. The whole BigBrain has been mapped for all the cortical layers. And then there's work, we have heard it here by Christian Schiffer and also many others, to perform brain mapping in the BigBrain. Also Kai Kiewitz and several other people have talked about this today. So that's what we have, and that's where we are. And when we zoom in into the BigBrain, we can even see some cells. So we are here, I think that is somewhere in the motor region, where we see really large cells, motor neurons, as individual black dots. But that is about currently the lower end of the resolution. So we can see some individual cells, but we cannot see every individual cell. Yeah? And there's some work, as we have seen, on super resolution methods, which drives this even further. But we are not at the stage where we see the individual cells here in the 3D model. And that's, of course, what we are after. We would like to have a model where we could perform quantitative measures of cells. And we would like to have a 3D model where we could do the really detailed cytoarchitectonic brain mapping that currently requires 1 micron sections. We have seen these works that employ Christian Schiffer's deep learning workflow, which goes into the 1 micron sections and then puts the results back into the 3D model. But it would, of course, be great to have a model at the 1 micron isotropic resolution where you could go into arbitrary oblique sections and perform mapping at the full 1 micron resolution there, and where you could extract individual cells in 3D. That would be interesting, and that's what we are after. And so there are, of course, a few things that we could do to help us. And one possibility is to add selected 1 micron samples to the BigBrain. And that is actually something that we have just done. So we have sampled patches from the 1 micron sections where we have a good quality and where we know in which brain region we are, and where we have manually annotated cortical layers. This has been done here with Sebastian Pludau and Casey Pacola. And then we detected individual cell bodies in these 2D patches using the method of Eric Upschulte that he has shown in the previous session. So this is a way, of course, to bring cellular detail in samples into the BigBrain model. And we are doing this. This is just about to be released for 10 different brain regions. And in each brain region, we have more than 10 different tissue samples, which are highly controlled. So these are high quality samples, carefully annotated. So that is a lot of work and far from being something automatic. But of course, this adds cellular detail. And this, you see here the points on the right, these are the samples of the patches that we have extracted. So there is a data set soon to be released that you can share. Then there is the work of Mona here that has been shown at the OHBM a while ago, where 1 micron sections have been aligned with the BigBrain model. And we talked a little bit about that we have 1 micron sections, of course, for this BigBrain model that we want to release. There's not every section, and some of the available sections have a poor quality, but many also have great quality, and we can do such an alignment here. Mona has shown that in her OHBM contribution, and this is of course difficult because if you go to the whole brain sections, it's large data, and you have to, as you see it here, you have to cut the images into tiles and work in parallel and so on. And Mona has managed this, and this will certainly be used to release such 1 micron whole brain sections in the nearer future. This would allow you to sample freely from the model. So it would be like less controlled than the previous approach, less curated, but more data, of course. What we would be interested in, though, is to really get 3D cell distributions. And we have done this for regions of interest already a few years ago. Here's a result from a publication that we did in 2018, where we have stacked 1 micron data, 1 micron sections in a subcortical region and extracted cell bodies and built such a 3D cell distribution here. But you run into a few problems if you do this with the 2D sections. So one obvious problem is you will have all the detected cell bodies aligned in 20 discrete 20 micron steps, because 20 micron is the thickness of the sections. And you could, of course, estimate the depth of each cell, for example, from the sharpness to get a more natural distribution. But there are more problems here because we have many bisected cells that appear in both sections. And for example, these cells would be double counted in such a case. And there are several problems with this. Also, we cannot appreciate the 3D morphology of the cell bodies. And there's a lot of work to be done repairing sections. There are some ideas to do this automatically, so you can read this in the paper here. But what we are currently trying to do towards a 1 micron isotropic model is to do Z scanning, so to subsample each tissue section, as you see here on the left, in different focal planes. This gives us a roughly 30 times increase in the amount of sections to be scanned. And we are going here from the terabyte size that the BigBrain has to the multi-petabyte size of data if we want to do that for whole brain. And that is an ambition. And what we want to do then on this basis is, and Marcel Heusegoms, who's certainly here with us, has also reported on this on OHBM, to really stitch such data for whole brain together in a way that we connect bisected cells, so that we can really do cell distributions in 3D that are much more correct than the previous work is. And Marcel has made good progress here on doing this, reconnecting bisected cells and using this to drive a registration of the whole brain. So basically to optimize the positions of those cells, fix the bisected ones, and arrive at a 3D reconstruction based on the work that Hartmut and Claude have just been presenting, which works on the level of images. This then goes to the level of cells and their constraints using the 20 micron work as a starting point. And this ends up in a really huge optimization problem if you want to do that for a whole brain. But we have currently done this for regions of interest as feasibility studies. You see here, for example, a part of the occipital lobe showing visual areas that we have anchored to the BigBrain. So this is, if we zoom into the BigBrain, the piece of the current BigBrain that you see, and this is what you can get if you do this Z scanning and such a way of reconstructing it. This is not yet perfect, but you see the much increased detail. But this is really huge data, then it's really difficult to do that. And we are progressing and we are on the way to do this. Here's another example in the motor cortex where we have done a smaller region with this technology. And what's interesting here, you can here see this here on the top left. This is the native tissue section. So this is really where the cut has been made, where we have a native resolution of 1 micron. The other directions are reconstructed sections that have never been measured in the microscope in this direction. And what we see here in the native section is we see here also motor neurons, but we wouldn't be able to perform brain mapping here because we don't see the laminar structure, because we are cutting here in a really very tangential angle. But when we go into the reconstructed section here on the bottom, which only comes available through the image registration, you see laminar structure, you see the cells, and this is where we could actually perform brain mapping. So this is where we are heading to, and this is of course difficult. And there are not only difficulties performing such a 3D reconstruction, but of course a lot around this in terms of image acquisition and data management. I have tried to visualize this here a little bit. So there is a whole lot of work to be done to do this high throughput acquisition for whole brain, to do quality control and metadata management. And we have a bunch of people working on these aspects. Then of course, the high performance computing processing of the data, object detection, 3D reconstruction, what we have seen already today. But then there's, of course, also the difficult question, how, if we ever have this data, how should we give you access to it? Because these are really petabytes. And there is actually a poster tomorrow by Pavel from our team, who is actually working on an image service that would allow to access such 3D reconstructions over the web, also over an API, efficiently, without us having to store the 3D reconstruction on disk, because we assume that this will be a highly incremental process. So this 3D reconstruction will certainly be repeated over and over with improved algorithms, with improved cell detections, with improved image preprocessing. And we cannot each time compute a new 3D model of a multi-petabyte size. So what we are aiming to do here is that we have actually the parameters of the reconstruction models stored and versioned, and also models for feature detection and so on, and that you can access the latest version just live by browsing the data without these images actually being somewhere on disk. Pavel can explain more about this tomorrow. And I think I used my time already, so I will end here. Thanks a lot, and I'm happy to take a question if there's time left.
I can't see any in the chat, but I have a brief question. I mean, all of that sounds amazing. You're having to reinvent every step of that to deal with such a large data set. Have you started to think at all about the next step? Because once you've created a data set, sort of with the creation of BigBrain, then people need to start creating the tools to analyze it. Have you started thinking about kind of the types of tools that might be necessary to do analysis on these multi-petabyte 1 micron data sets?
Of course, there are some, yeah, and I think many different people have many different ideas how to analyze it, and that's what they should do, yeah. Obvious things are detecting cells in 3D, clustering cells, things like this, and there's of course work in different labs on such aspects that we would like to enable. I see two major ways of doing that. One way would be to download regions of interest from this huge model, and we can do this. You have seen the 3D viewer for the BigBrain, and I imagine that you could then, for a region of interest there, when you zoom in, just download the chunk, and there are ways of doing that. I have shown some of these ideas actually yesterday in the hands-on sessions, and then do the analysis on your side. But there might be people who want to do really big data analysis. And in these cases, it will certainly be necessary to send the algorithms to the data. And we have yesterday also heard from Tristan Glattard and Maurice Riedel and other people how such workflows could be organized, yeah, so that you could send algorithms to data using tools like CBrain, DataLet, and using the HPC infrastructure. So I think both ways will make sense for different types of studies.
Great. Thank you very much, Timo.